Overview of the use of Murine models in leukemia and lymphoma research. The applications of Murine models of cancer are numerous: mechanism discovery, oncogenesis, molecular genetics, microenvironment, metastasis, and therapeutic efficacy. Leukemias and lymphomas are a group of highly heterogeneous hematological malignancies that affect people of all ages and ethnicities. Leukemia and lymphoma arise from hematopoietic and immune cells and usually spread widely throughout the body. The liquid nature of many of these malignancies, as well as the complex microenvironment from which they arise and their multi-phase genetic basis, has added to the difficulty in generating appropriate and translational models to study them. The mouse as a model. The mouse is now widely considered the model organism of choice for the study of human disease. The reasons for this choice are numerous, including relative genetic similarity of mice to humans, their small size, reliable breeding, and short lifespan. Mice also share many physiological characteristics with humans and therefore provide similar organ system biology for the study of cardiovascular, endocrine, immune diseases, and others. Concerning hemopoiesis in mice, lymphocytes are the predominant circulating white blood cell, whereas the neutrophil predominates in humans. Bone marrow in the mouse retains abundant hemopoietic components throughout life in contrast to the polycellular marrow of adult human. Choosing the correct model. Murine models are abundantly plentiful commercially and as a standard of research are often easiest to cite and utilize previous work to develop your model. One early and critical consideration is the selection of the strain of mouse. The high incidence of leukemia and lymphoma in some mouse strains is further enhanced in immunocompromised mice as an experimental tool often used to model these diseases. Some spontaneous leukemias in particular can be difficult to distinguish from the disease model manipulation. It is clear that many of these conditions could confound individual studies, and therefore the strain of mouse must be considered carefully. Develop mouse models and their utility. The genetically engineered mouse model has been used to mimic many human cancers with an etiology based in genetic aberration. To produce transgenic mice, a gene of interest in the form of a vector is injected into the fertilized egg. The resultant of offspring will carry additional copies of this transgene. So here we have in figure 1 a xenograft model. In A we have the cell line derived cells, and in B we have the patient derived cells, and these can be transplanted into an immunocompromised mouse, which is used to study several aspects of tumor biology and behavior, such as the rate of growth, biologic behavior, biomarkers, and the response to drugs. Murine models of leukemia and lymphoma. In leukemia, Murine models have been instrumental in elucidating immune reaction, hematopoietic stem cell niche and microenvironment, cancer stem cells, novel therapeutics, and chemotherapeutic resistance in acute myeloid leukemia. In lymphoma, humanized mice infected with the Epstein-Barr virus develop a B-cell lymphoproliferative disease and EBV-associated hemophagocytic lymphohistiocytosis. The use of these models has also elucidated the role of innate immune responses, including EBV-specific adaptive T-cell responses in these diseases. EBV-associated Hodgkin's lymphomas developed in mice with activated T-cell environment, while non-Hodgkin's lymphoma developed in T-cell depressed mice following infection with EBV. So, in conclusion, development of translational Murine models have been vital to the detailed mechanistic investigation into disease pathogenesis and identification of therapeutic targets. The mouse provides a valuable model system to study human diseases in its genetic and physiologic similarity while being readily available and economically practical. Although the mouse's reign as king and biomedical research will likely continue for many years, appropriate model design throughout characterization and excellent new technologies will allow researchers to strive further to replace, reduce, and refine their use.